today we're exploring CRISPR-Cas9, a precise tool for genome editing. Introduction to CRISPR-Cas9. Think, what if scientists could now edit human DNA just like you edit a Word document? They can find, replace, delete, and insert. But instead of fixing spelling mistakes in text, they're fixing genetic mistakes that cause terrible diseases. This isn't a movie anymore. In December 2021, a 13-year-old girl named Alyssa became the first person ever cured of a deadly cancer called leukemia using this technology. Her T-cells were edited at the gene level to become superfighters against her cancer. 6 months later, her cancer was completely gone. The technology that made this amazing cure possible? CRISPR-Cas9. CRISPR-Cas9 is a precise gene editing tool that uses guide RNA and the Cas9 protein to target and cut specific DNA sequences. It can repair, remove, or replace genes with applications in treating genetic diseases, research, and biotechnology. Its accuracy and simplicity make it a revolutionary tool in modern biology and medicine. By the end of this video, you'll know exactly how it works. One letter in 3.2 billion. How tiny DNA errors cause massive human disease. Let me show you just how huge this revolution really is. The human genome has 3.2 billion DNA base pairs. That's like a book with 3.2 billion letters. If you printed this book, it would be 262,000 pages long, or about 1,000 regular-sized books. Now imagine that changing just one single letter, 1 out of 3.2 billion, could cause cystic fibrosis. Changing one other single letter causes sickle cell disease, which hurts 300,000 newborn babies around the world every year. Another single letter changes causes Huntington's disease. There are over 10,000 known single-gene disorders, affecting 1 in 300 people worldwide. That means 25 million people just in the United States are living with genetic diseases caused by these tiny DNA mistakes. From Mendel to CRISPR, how gene editing finally became possible. For more than 100 years, ever since Gregor Mendel discovered how traits pass from parents to children in 1865, scientists dreamed of being able to rewrite DNA. The Human Genome Project, finished in 2003 and costing $2.7 billion, let us read DNA for the first time. But reading and editing are completely different things. Before CRISPR, genetic engineering was like doing surgery with a sledgehammer. Rough, expensive, and often didn't work. It could take months or years to edit just one gene, costing hundreds of thousands of dollars. Then, in 2012, 2 scientists published a paper that changed everything. Jennifer Doudna and Emmanuel Charpentier showed how to turn a bacterial immune system into a programmable gene editor. Suddenly, what used to take months could be done in days. What used to cost a fortune now costs less than $100. Outline of today's lecture. CRISPR-Cas9. Explained. In the next lectures, I'm going to take you on an amazing journey from the tiny world of bacteria to modern hospitals where genetic diseases are being cured. You'll learn exactly how CRISPR-Cas9 works, how it finds its target among billions of DNA letters, how it makes its cut with perfect precision, and how scientists control what happens next. We'll look at real patient stories, including Victoria Gray, who hasn't had a sickle cell crisis in over 1,400 days since her CRISPR treatment. We'll explore the numbers, why this technology works 95% of the time in some cases, and how it's being used in over 300 clinical trials around the world. Whether you're studying in medical college or university or just curious about the future of medicine, This complete guide will teach you everything you need to know about CRISPR-Cas9. In Biomedica Explained YouTube lecture series, we make complicated science easy for everyone to understand. Let's start. The microscopic arms race: how bacteria invented CRISPR to survive viruses. Our story starts in the invisible world all around us, where bacteria and viruses have been fighting a war for 3.8 billion years. Every single drop of seawater has about 10 million viruses and 1 million bacteria. That's more than all the people on Earth in just one drop of water. These viruses, called bacteriophages or phages, are amazingly good at killing. A single phage can inject its DNA into a bacterium and in just 30 minutes make 200 new viral particles that burst out of the cell and kill it. With numbers like that, you'd think bacteria would have disappeared long ago, but they haven't. Bacteria make up about 15% of all living things on Earth. How do they survive? The answer is CRISPR. The discovery of CRISPR: how bacteria keep genetic records of viral invaders. Finding CRISPR is like a mystery story. In 1987, a Japanese scientist named Yoshizumi Ishino was studying a totally different gene when he saw something strange: repeated DNA sequences with unique spaces in between. He had no idea what they were for. In 2002, a Dutch scientist named Ruud Janssen gave these sequences the name CRISPR, clustered regularly interspaced short palindromic repeats. Still, nobody knew what these sequences did. The big discovery came in 2005 when 3 different research groups found something shocking. Those spacer sequences weren't random junk DNA. They were perfect matches to viral DNA. The bacteria were keeping genetic photos of their enemies. How bacteria remember viruses using CRISPR. Here's how this bacterial immune system works, and it's absolutely brilliant. When a virus attacks, the bacterium has about a 1 in 100 chance of surviving the first attack. But if it does survive, it takes a sample, a 30-nucleotide piece of the attacker's DNA. This sample gets put into the CRISPR array, like adding a new photo to a criminal database. The typical bacterial CRISPR array has between 20 to 50 of these viral photos, showing a family tree of past attacks that could span hundreds of years. When the same virus attacks again, the bacterium is ready. It copies the stored sequence into guide RNA and pairs it with Cas proteins. How well does it work? An amazing 87% protection rate against repeat infections. How scientists reprogrammed CRISPR to precisely edit any gene. The aha moment came when scientists realized they could steal this system. Instead of letting bacteria choose which DNA to target, researchers could design their own guide RNAs to target any sequence they wanted. It was like reprogramming a guided missile system to hit any target you choose. The possibilities were mind-blowing. Any gene in any living thing could potentially be edited with perfect precision. This realization started the CRISPR revolution that's still changing science and medicine today.